All right. Today I want to talk about another mutation mechanism. This is simple sequence repeats. So a simple sequence repeat is basically a type of repetitive DNA where you basically have either one or a few DNA letters that are just repeated over and over again. Sometimes there's one, sometimes there's two, sometimes there's three or four, but these are basically very simple sequences that are repeated in the DNA. And so what's interesting about this is that these things allow for actual tuning knobs to be present in the genome. And so a group that's done a lot of work on this is Kashi and King. And so this is the first paper that I read on the subject. Simple sequence repeats as advantageous mutators in evolution, and you can find those. You can find those two. Have written a lot of different papers on simple sequence repeats in evolution, and uh, it's also present in the book The Implicit Genome, which I recommend to all of you. It's a great book. So anyway, simple sequence repeats. So what's interesting about simple sequence repeats is that they are some of the most mutable categories of DNA, and they mutate almost entirely by changing copy numbers. So rather than actual DNA base pair substitutions, they almost entirely mutate by copy number variations. Okay, and so because of this, you can see that so you have identified segments which are highly mutable because they're sequence repeats, and they mutate in a specific way by lengthening or shortening. Okay, so you basically have these tuning knobs. So it's basically allows the DNA itself to kind of adjust the length parameters. Um, and so there's a question whether or not this is in response to selection or not, and that's a good question. But it's interesting that it seems that the DNA itself is pointing to likely locations where mutations are worthwhile. What's also interesting is I've actually found, I don't have the reference for this on me, but cancers oftentimes happen when a single base pair mutation makes a segment that was not previously a simple sequence repeat look like a simple sequence repeat to the genome. So you have a single base pair change, which now makes something look like a simple sequence repeat, and so the genome um, will start making more copies of that, more or fewer. And that oftentimes leads to cancer. And so it's really interesting because when you have these simple sequence repeats, that that most of them are very functional. The few that turn to cancer are usually because they used to not be simple sequence repeats, and some sort of a DNA mutation caused them to look like simple sequence repeats. So we have a sort of degeneration in the genetic code will cause those things to lead to cancer. So anyway, so what this allows, it can do a lot of different things. It can turn genes on and off. So for example, you can have, if something is not uh, uh, in multiples of 3, so if you have like one that's multiples of 4 or something, having another copy might turn the gene off. So for example, it might cause something to turn into a stop codon. It'll definitely change the sequence of things afterward because everything's going to now have a new starting codon. It's going to shift it, do a frame shift of the DNA. And then so it can turn genes on and off. It can also it can affect the regulatory regions. So it can affect what binds to the regulatory regions. It can also be used to adjust in non-coding regions. It can be used to adjust gross morphology or gross morphology um, or biochemical. So like having a more distance between the promoter and the actual gene might decrease the amount of transcriptional activity. So it might repress to some extent the outcome of a biochemical process. So you can see that these things can be used to tune parameters. There's also another paper called Molecular Origins of Rapid and Continuous Morphological Evolution that talks about these. And so, for example, now just so you know, simple sequence repeats actually go by a couple of different names. So um, the in this case, I think they use a different term. What is it? I forget which term these guys use. Tandem repeats is what they call them here. So anyway, one example that they show is the bend in a dog's nose is actually based on repeats. And so you can have different dog breeds that arise, because that's why we have rapid evolution of dog breeds, because uh, uh, you have these segments of repetitive DNA that evolve almost entirely by copy number variation, and these copy number variation are basically tuning knobs to the parameters of dog. And so these parameters generate morphological changes, and that generates your different dog breeds, and that's why they that's why they're able to generate so rapidly, is because it's just a copy number change of a tuning parameter. So anyway, so that's simple sequence repeats. There's also another one that I haven't looked into as much as I wanted to. It's there is an antifreeze protein. I wish I had the link for you. There's an antifreeze protein that I believe is just an expansion of a repetitive sequence, and I haven't looked into that enough to know because it looks like that what's happened is that as this grows and repeats, basically allows it to be more and more antifreeze. And so anyway, you can imagine something adapting to colder climates by expanding a repeat section. So anyway, I'll see if I can, actually I might be able to just Google that. Antifreeze protein um, repeats. So let's see here. So we have evolution of hyperactive repetitive antifreeze proteins in beetles. This might have, I was actually thinking about fish, but let's see here. They're frequently encoded by tandemly arrayed multigene families, so that's not what we're actually looking at. They show variation in number of repeats. So that's, anyway, so I've always wondered if the repeats were related to the amount of antifreeze that was available. And it's something that, I don't know the answer to that, but it's something that's that would be interesting to look into. Let's see here. I think the one that I was actually looking at was fish. Yeah, so the glycoprotein antifreezes, which are made up of a repeating tripeptide. All right, so you have this repeating tripeptide, and so I've always wondered if the amount of repeat, so as something adapts to more and more cold, if it can expand this repeated element. So anyway, again, a question, but it's worth looking into, and also is kind of an example, not out of bounds for what we've generally seen in biology. So anyway, simple sequence repeats. Check out this article, Simple Sequence Repeats as Advantage Mutators in Evolution.